Hi, my name is Kabishiro Mulligay, and I'm a grade 10 student at Fort Richmond Collegiate, and I live in Winnipeg, Manitoba, Canada. My science fair project is Battling the Beast, Biocontrol Interventions Against Fusarium Graminearum to Control Fusarium Head Blight. Fusarium head blight disease is a fungal disease of various grasses that stops kernel growth, which is often found in wheat, but also affects barley, oats, rye, and other forage grasses. This disease is very common throughout the prairie provinces, which spreads throughout Manitoba, Saskatchewan, and Alberta. The first severe outbreak occurred in Manitoba in 1993, which was caused by the several fungal species of Fusarium genus. Fusarium graminearum is a species which causes the most severe damage to crops. Fusarium graminearum is a fungal plant pathogen which invades plants and grains, mainly wheat spikes. The fungi appears as a reddish-brown fuzzy flower on PDA plates. It often grows between the ranges of 5 to 30 degrees Celsius, with the optimum growth being obtained at 25 degrees Celsius. Fusarium head blight is the causing effect to the reduce in farming yields. This disease affects biofuel production, baking and the milling of wheat, malting barley, and brewing of qualities of mulled barley and livestock feed. Through past research, it was found that the Pseudomonas and Bacillus species are capable of secreting antifungal metabolites that prevent pathogen infection. In the latest studies, this biocontrol agent has been able to control fungi such as Brassicus napus. This project helps to prove the effectiveness of different biocontrol methods to reduce the growth of fungi. 3 different bacteria were tested: Pseudomonas chlorophyll, Pseudomonas brassicacearum, and Bacillus liquefaciens, against 4 different strains of Fusarium graminearum, which were found throughout Saskatchewan and Manitoba. In this experiment, LBA agar plates were used to grow the fungi and bacteria. The growth of the fungi over a period of 10 days was tested by measuring the diameter of it. These results were compared to the control group of fungi with no biocontrol agents. My hypothesis of my experiment was that biocontrol agents do have a significant effect on controlling the growth of Fusarium graminearum, and my null hypothesis was that there is no significant effect when using the biocontrol agents to control the growth of this fungi. And my objectives were to find out the effectiveness of biocontrol agents in controlling Fusarium graminearum as a solution to prevent Fusarium head blight, to find out which bacteria species has the highest effect on controlling the growth of Fusarium graminearum, and to find out if the experiment is repeatable by repeating it 4 different times with 4 different fungi strains. Now, these are the materials which I used. I used a total of 96 LBA plates, an inoculation needle, a cork borer, a loop, 90% ethanol, Q-tips, a laminar flow cabinet, a Bunsen burner, 4 different fungal cultures. 2 were from Saskatchewan and 2 were from Manitoba, the bacterial cultures, a lab notebook, a marker pen, and a ruler. To start off with the procedure of my experiment. So before I started off with doing anything in my experiment, I made sure to follow aseptic techniques which were applied prior to the experiment. And doing this prevented microbial contamination, and it allowed my environment to be disinfected so that I could get the most valid results. I started off by setting up my LBA plates, and I used 24 LBA plates for each fungal culture. 6 were used for P23, 6 were used for DF41, and 6 were used for B56, and 6 were used as the control. I then divided the bottom of each plate into 4 quarters using a pen, and then I marked lines on each test LBA plate, which will be streaked with bacteria. I repeated these steps for each fungal culture. The fungal transfer and inoculation. So I started off by extracting 12 discs from the edge of the fungi culture plate using a sterilized cork borer. I sterilized the needle with 90% ethanol, and then I followed the same sterilization steps, which was dumping it into 90% ethanol and then burning it until it was red hot. I gently picked up the fungi once the cork borer and the needle was cooled down. I picked up the fungi from each disc, which was obtained from step 3.1, with the sterilized needle, and I transferred the disc into the center of the LBA plates using the inoculation needle technique. 1 disc was put into each LBA plate. Next, I did the bacteria transferring and the streaking. I took a sterile cotton swab and I swept it back and forth on the surface of the bacterial culture, and I used a separate cotton swab for each transfer. Using the cotton swab, which has the collected bacteria on it, I transferred a portion of the bacteria to each of the test plates using the streak inoculation technique. And I left the plates on the shelf underneath the light bulb, which was 57 watts. And I repeated these steps for each of the bacterial cultures. Days allowed the fungi to start growing, and I started to measure the diameter of the fungi every 2 days until 10 days was over. I used a ruler and placed it on the bottom of the plate on the horizontal line which I marked before, and I measured the diameter of the fungi growth. And I did this one more time by placing the ruler on the vertical line once again, measuring the second diameter of the fungal growth. I recorded the measurement and the observations, and I repeated these steps for all the other plates, which showed the growth of the fungi. And I repeated this experiment for each fungal strand. So for SK7, SK15, M114, and M118. So here are a bit of observations that I received. So over here are the plates on day 1. And as you can see, I had multiple samples of my plates. The next picture is my plates on day 2, and you already can see the fungi growth. But when you compare it back to the control in that picture, you can see that the bacteria is stopping it as it is a biocontrol agent. And on day 4, you can already see that the control fungi is starting to grow so much larger than the fungi that are being controlled by each bacteria type. So these are the final results of my experiment. And on day 10, this is the Pseudomonas chlorophyll, which is the orange fungi, then the Pseudomonas brassicacearum, and the Bacillus liquefaciens. And as you can see, right next to each bacteria culture with the fungi in it, there is a control group of the fungi. And it is quite obvious to see that the control has grown to its full extent and the full diameter of the plate to the point where the control started to grow vertically because it had no more space to grow. As when you compare it to the fungi, which is being controlled by the bacteria, we can see that it is completely being controlled, and the fungi actually starts to grow in diagonal ways, trying to escape the antifungal metabolites, which the bacteria is secreting into the agar to stop the fungi growth. So when you look at my results, this graph shows the average fungi growth of SK7, which was the Fusarium Gramineearum, which was from Saskatchewan, against the 3 different bacteria species. So in this graph, you can see that the control had an average diameter of 8.50 centimeters. The control always having an average diameter than the biocontrol agents and the biocontrol plates. It always had a higher average because immediately after day 2, you can see that it already spiked to its maximum growth. Now, the Pseudomonas species, which is PA23 and DF41 at 10 days, show that the control had an average diameter of 8.50 centimeters, while the Pseudomonas species had an average diameter of 5.38 centimeters and 5.95 centimeters. And the Bacillus species at the end of the day had an average diameter of 5.07 centimeters, which was the lowest diameter growth of the fungi that was observed with this fungi strain. Now I obtained similar results for each graph, so I will just give you a brief overview of each. So for the SK7 fungi, the biocontrol agents all maintained a similar diameter growth throughout the 10 days, which was ranging between 4 to 5 centimeters. And as you can see, the control maintained the same diameter growth as in the last strain, which was 8.5 centimeters, which was the full extent to which it would be able to grow. These next ones show the fungi strands from Manitoba, and you can see the results are the exact same with just slightly different measurements. For the first one, which is M118, you can see that the control, once again, grew to its full extent of 8.50 centimeters, while the Bacillus species actually ended up having the lowest diameter growth once again of 4.98 centimeters, and the Pseudomonas brassicacearum and Pseudomonas chlorophyll species had a growth between 5.5 centimeters and 5.9 centimeters. And these results were also similarly attained to the M114 fungi strain. So to analyze my data, I used ANOVA single factor. So using the statistical ANOVA single factor analysis, all my results show that the biocontrol agents do have a statistically significant effect when controlling the growth of Fusarium Gramineearum. This is due to the p-value being less than or equal to 0.05, and that shows that my results are statistically significant. and also having my F value be greater than my F critical. Having my results pass both of those tests in the ANOVA single factor, this proves that my hypothesis is proven and my null hypothesis is rejected. To restate, the purpose of my investigation was to test the effect of the specific biocontrol agents in preventing the growth of Fusarium graminearum. When being exposed to 3 different kinds of bacteria, the diameter growth of the fungi was measured over a period of 10 days and was compared back to the control after the data was collected. So, in conclusion, my hypothesis was proven, and biocontrol agents do have a significant effect when controlling the growth of Fusarium graminearum. And according to ANOVA, the ability of the biocontrol agents to control the growth of Fusarium graminearum is statistically significant, since my null hypothesis was rejected. The Bacillus bacterial species saw the highest effect on controlling the growth of the fungi when you compare it back to the Pseudomonas species. The lowest diameter growth of the Fusarium graminearum was obtained in the plates with the biocontrol agent of Bacillus emoli liquefaciens. The repeatability of my experiment was also proven, since the similar results were obtained with all 4 different fungi strains of this fungi from Saskatchewan all the way to Manitoba. So now for my applications and my implications. These results can be used as a sustainable plant disease management strategy by introducing these biocontrol methods to farmers. It can be applied as the most appropriate and effective alternative to chemical fungicides and to minimize the environmental pollution caused by chemical applications. It would have a huge positive effect to the decrease in the disease of Fusarium graminearum and the disease of Fusarium head blight, as it would decrease the number of numerous outbreaks which kills crops and livestock. This information can also be used to further other developments in science by applying it into the research of mycotoxins of Fusarium graminearum. The most common mycotoxin in this fungi, which is associated towards Fusarium head blight, found in wheat, is DON. Biocontrol agents can be implemented into the large stalk of wheat, which will decrease the presence of mycotoxins since Fusarium graminearum is being controlled. This is beneficial towards livestock, protein synthesis, humans, and the increase in feed intake. This project can also be applied to genetically modify wheat to already have those biocontrol agents installed in them, so we don't need to spray it directly onto the plants. And what's next? Some further questions that I have and some further research questions that can be applied to new projects or an extension to this project is, how can this research be implemented with other fungal species? How does this affect the growth of the canola, wheat, and barley plants? And how does the application of the biocontrol agents to the wheat spikes affect the microenvironments which are exhibited in the soil and the grass? And lastly, I would like to say my acknowledgements. I would like to express my most sincere gratitude towards Dr. Delunthor Fernando, which is a professor of the Department of Plant Science in the University of Manitoba, for making this research possible and providing me with lab space at the University of Manitoba. This allowed me to conduct my experiment. I would also like to show immense gratitude towards Monica Dariasna, who is a postdoctoral fellow in the Department of Plant Science at the University of Manitoba, for assisting me throughout this project, teaching me the basics of working with fungi and bacterial cultures, and providing me with the specific fungal and bacterial cultures that were needed to perform this experiment. And lastly, I would like to show my appreciation towards my family for encouraging me during this entire process. Thank you, and I really hope you enjoyed my presentation.